Hey everyone, welcome to Apology with Mr. Jennings. Today we're going to talk about meiosis. Just a little bit of a review here. Meiosis was back a couple weeks ago, just kind of recap some things. Again, as I mentioned in the previous video, if we're following these in order, you already have this as a note handout. So just follow along. I'm mainly just going to elaborate just so you have a record to go back to and talk about. Because we really didn't get to complete our last unit in class, so we want to make sure we have that. So let's talk about meiosis here. Now, meiosis is the process of producing sperm and egg cell from cellular division. It is very similar to mitosis. Terminology is very similar. However, they are not exactly the same thing. Big thing is there's two stages. It's a reductive cell division, and it produces 4 daughter cells that are not identical. Meiosis, though, goes from interphase 1 to prophase 1 to metaphase 1, anaphase 1, and telophase 1 in cytokinesis. And each phase, let's see if I got through it all. Alright, so interphase 1 is basically regular interphase. But when you get to prophase 1, each chromosome pairs up with its corresponding homologous chromosome to form a tetrad. Remember, tetra means 4, so they're going to form tetrads. Now, during tetrads, there's going to be an event here called crossing over, and we'll talk more about that. Like metaphase 1, the spindle fibers will attach like they would in regular metaphase, and they're going to then go into anaphase 1, where the spindle fibers are going to separate the homologous chromosomes, not the sister chromatid, but the homologous chromosomes. And then in telophase 1, we officially entered what we call a haploid state, and the cells are now have half the DNA. Now, in a haploid state, they're then going to go into meiosis 2. In meiosis 2, they're going to go to prophase 2. Now, prophase 2 is going to result in 2 haploid daughter cells in meiosis 1. Meiosis 1 is going to have 2 haploid daughter cells, and each half of those are going to have only half the regular DNA. So the spindle fibers are going to reform yet again. They're going to attach as the sister chromatids line up down the middle of the cell, and in anaphase 2, they're going to be separated. They're going to be pulled apart. The sister chromatids are going to separate and move towards opposite ends of the cell. And in telophase 2, you're going to have 4 haploid daughter cells that are genetically different. For instance, here, now let's talk about the other stuff. Oocyte or spermatocyte, so we're going to talk about, is it a male or female sex cell? This cell undergoes meiosis originally, has 6 chromosomes, and is replicated to form 12 chromosomes in preparation for meiosis. Set. Now, prophase 1's, the homologous chromosome pairings, again, into tetrads. Just kind of recap some stuff here and see a little bit up close. So as they form these tetrads, they're going to then separate in metaphase 1s. They're going to line down the metaphase plate, as we see here. In anaphase 1s, the homologous chromosomes are going to separate. This is where the first non-disjunction event could occur. This is when they have a failure to separate. This is what's going to lead to trisomy and monosomy disorders. As the 2 sets of chromosomes, the homologous chromosomes fail to separate in case, they'll go basically one cell will be lacking a chromosome and the other cells will be with a chromosome. In telophase 1, membranes are going to form around the separate homologs. Then we're going to go straight to prophase 2. The big thing is we do not go into another interphase. So DNA is only replicated once in interphase 1. Now, as we go from telophase 1 to prophase 2, no DNA replication has occurred. So we only have half the DNA. The spindle fibers are going to start binding the sister chromatids. They're going to line up down the middle of the metaphase plate. The sister chromatids are going to be separated and move into opposite poles. So the X's break down into individual lines. And in telophase 2, nuclear membranes are going to form around the sister chromatids, the separated chromatids. And notice, each cell only has half the amount of DNA as the original cell. That's why they're called haploid. Actually, all the cells in meiosis 2 are haploid. And telophase 1 is also haploid. And here's an example of a non-disjunction. Non-disjunction in meiosis 1 versus normal meiosis 2. All the offspring are going to have 100% abnormal number of chromosomes. In meiosis 1, if there's a normal meiosis 1 but a non-disjunction in meiosis 2, in anaphase, for instance, in anaphase, it can have another non-disjunction. 50% of the gametes produced will be abnormal, the other 50% will be normal. So it depends on where the impact occurs. If it occurs in meiosis 2, the impact is mitigated. The If it occurs in meiosis 1, everything downstream from meiosis 1 is affected. Now, this brings up another question. How can siblings, knowing that your parents' DNA is half, you have half your mom's, half your dad's DNA, how's it possible that they do not look exactly the same, but they look similar enough that you know they came from the same parents? Well, a big part of that is crossing over. Another part is you have independent assortment. You get it random, you get independent assorted chromosomes from each parent. This is one of the Mendel principles. But another major factor is crossing over. See, during prophase ones, when they form tetrads, there's times when the chromosomes are going to overlap on each other. As they overlap, they're going to break apart. And when they separate in anaphase 1, they're going to have completely mismatched DNA. This creates a combination of different traits. So the gene combination you get from your person gets from the parents will be different to varying degrees. And then they may get a combination that's different from the sibling. This is also how you can get some traits that are normally considered linked, found on the same chromosomes we discussed last week, in a non-combined form. So you can get unlinked traits, to unlink them by crossing them over. So for instance, commonly known blonde hair, blue eyes is considered a linked trait. They're found on the same chromosome, the genes necessary for blonde hair and blue eyes. Crossing over allows for combinations like red hair, blue eyes, or brown hair, blue eyes, or even blonde hair, brown eyes. And that is all because of the crossing over. So whenever those chromosomes line up, if they were the same genes on the same chromosome, they could sometimes get randomly mismatched. And that's actually going to be a very big driver of genetic diversity. And there's some more sibling similarities here. So, meiosis is the process of creating meiosis is the process of creating sperm and egg cells, gametes. It's necessary for sexual reproduction. We've already talked about this a little bit. We did a poggle on it and everything. Now, the combination of sperm and egg is called fertilization. And depending on the chromosomes, basically, meiosis is going to be a way of orderly dividing up those chromosomes. By altering the chromosome count, you can create it through nondisjunctions. You can create various trisomy and monosomy disorders that we discussed in the previous. And the genetic disorders you're going to research this week are examples of these, nondisjunction events, in addition to other things. All right. That said, next time we're going to come back, we're going to talk about how chromosomes themselves don't necessarily have to be nondisjunction. The chromosomes themselves can mutate and get rearranged, and those are called chromosomal disorders and mutations. That's all for today, though. I've been Mr. Jennings. Hope you're having a great time. See y'all soon.